All right, so questions on the study guide for Chapter 3, Part 1. Remember, you're only supposed to know, or expected to know, the bold items on there. Yes. The functions of each of those? Yeah. So, because probably a lot of you wrote a lot of stuff on there. So plasma membrane, it basically lets things in and out of the cell. That's its main function. Yeah, let's finish these first. Oh, sorry, there's all of them.
Oh, sorry.
That's fine. So then ribosome makes proteins. The rough ER sorts proteins. The smooth ER does all kinds of different things in different cells, makes lipids, detoxifies, stores ions, would be 3. It makes lipids, detoxifies, and stores ions. The Golgi there, it packages up proteins for export. It puts them in vesicles. The lysosome, I call it the recycling center. So it breaks down old, worn out stuff in the cell so it can reuse the parts. And who knows the mitochondria? It's the powerhouse. There was that thing on Facebook a year ago. Only thing I remember from biology is the mitochondria is the powerhouse, which means it makes ATP, energy. And then you want to talk about transcription and translation?
Yes.
Please, Edward. So we're going to talk about in more detail a little bit later today, but purposes of the quiz, this is what you want to know. So basically, to go from DNA to protein, there's 2 steps. So DNA to mRNA is transcription. And then mRNA to protein is just translation. And the way you remember which one's first, because trans is the same, right? Then you look for the next letter. So C comes before L. That's how you can keep them straight.
When it says in the translation, it says basically the small definitions where it said it's nucleotides to amino acids.
Mm-hmm.
So that's nucleotides to amino acids, and amino acids turn to protein, right?
Yeah, so a protein is a sequence of amino acids. Mm-hmm. DNA is made of nucleotides. RNA is made of nucleotides. Protein's made of amino acids. Yeah, and we'll talk about the central dogma in the lecture. more details of why it matters. And yeah, just know for the quiz at least, just that. It's good enough. All right, you ready for your quiz? You asked me about all the study guide, which is fine. I'm glad. Basically, chapter 3 is how cells work. So it's like a whole intro bio class in one chapter, which is why I split it up.
With a bunch of words I never heard.
With a bunch of words you've never heard of. Exactly.
That I don't, or that I didn't remember.
All right, so trying to keep it kind of simple for most of it. So cell cycle is the process cells go through to reproduce and make new cells. And the main phases are G1, where the cell grows, S, where you replicate your chromosomes, because if you're going to take one cell and turn it into two, you better make sure those two new cells have the same DNA as the first cell. So that before you get too far, you better copy it. So that's all S phases. It's copying the DNA, copying the chromosomes. G2 is some more growth. And then M phase is mitosis or meiosis, which we'll talk about in a little more detail. But those are the two processes of cell division, where the cell actually divides into two. And they're similar, but not.
Is the amount of time it takes each phase important?
No. And that's because there's no, like in your body itself, all your cells are going through the cycle at a different speed.
They just have that time frames and everything.
Yeah, that would be like in culture, if you're feeding them lots of nutrients and telling them to grow and grow, that'd be like the fastest they could go. In general though, I mean, I would say the mitosis stage is very short. The part where you're actually dividing is very short. Those other three phases are a lot longer. If you want some extra review, there's a fun little game you could play on your computer. It'll get you acquainted with all those phases, and that's actually pretty fun. All right, so we mentioned you've got mitosis and meiosis. Start with meiosis. So meiosis has one function. It's to make sperm and eggs. That's it. So that's only in the ovaries and the testes. Mitosis is when you need cells, new cells for everything else. So you saw in that little fun facts things, what was it, 200 million cells die every day? So they're replaced by mitosis. So for example, your skin, like, think how tiny a cell is. You know, you looked at basically a skin cell from your cheek in lab. Think how many of those cells are on your skin, and they're not really held there too tightly. They're always just falling off. Same thing with your intestines, your esophagus. All throughout your body, the cells are dying, and then the cells underneath them or next to them divide to replace them. So that's replacement cells. When you were just a little fertilized egg, that was one cell. That cell divided by mitosis trillions of times to turn you into a person. So that was all by mitosis, going from one cell to the trillions of cells you have now. And wound repair, like if you get a cut or something, they'll repair that way. Or if your liver gets damaged, the cells can go through mitosis to make new liver cells to replace the damaged ones. So one important thing to note is the numbers of chromosomes that we're talking about. So obviously we have 46 chromosomes, right? 23 pairs. And so for mitosis, get this off here. So for mitosis, you start with one cell that has 46. That's in G1. And then after S phase, how many are you going to have? What does S phase do?
Oh, replication.
So if you had 46, you copied them all. Now we have 92. Then after M phase, we're going to split this cell in half. So now you have 46 again. So same as what you started with, except now you have 2 cells of it. So that's 1 round of replication, 1 round of division for mitosis. And then for meiosis, kind of how many chromosomes are you starting with in a meiosis, do you think? Starting with. You are starting with the same number. We are eventually going to get to 23, though. What about after S phase in meiosis? What does S phase do? So 92. And then M phase, we've got 2 rounds of cell division. So how many cells are we going to end up with? So if we split this cell once, make it a little bigger here. And then we split it again. Now we have 4 cells. How many of each of these cells have? That's where you get your 23. So this could be sperm or eggs, depending on if you're a man or a woman. So now we have 23, which is good. Those are called haploid cells. These are diploid cells. So haploid means half the number of chromosomes. All right. So first question for the day. At the end of meiosis, what do you have? All right, so most people chose B there. The answer is actually D. So all of these cells do have 23 chromosomes, but it's a random mixture of the chromosomes you got from your mom and your dad. So this has different chromosomes than this one, than this one, than this one. And that's where you get a lot of your diversity from.
If the cells that are dividing meiosis, that like your sex cells that are dividing, how do they start out with 46 chromosomes and if your sex cells only have 23?
So to make sex cells, you start with normal diploid cells of the body. So you have specialized cells in the ovaries and the testes that will eventually turn into the sperm or the egg cells. And when you get to 202, we talk about this process in a lot more detail in the reproduction chapter about how you go from here to there.
Can you explain a little bit between like we all thought it was like the 4 genetically identical daughter cells?
Mm-hmm.
I'm getting kind of confused on like how it's non-identical.
Okay, yeah, so let's pick out, tell me some traits that people have.
Eye color.
Okay, so tell me some eye colors though. Brown, blue. Okay, well, three's enough for eye. What about some other traits? So these are eyes, hair, brown. What's something else? Okay, so height. So basically for each of these traits, you have 2 alleles for them, right? You got one from your mom and one from your dad. So maybe you got 1 brown eye allele, 1 blue. Maybe 2 red for hair. So 2 of each of these. When you make your sperm and your egg, you randomly pick 1 from here, 1 from here, 1 from here.
The chromosomes split in half.
Right.
Right, okay.
So the S phase is just a big pile of,
Big pile of chromosomes, big pile of traits.
and they just,
Um,
and then that's when the dominance occurs, and then it makes like your.
so the dominance is when you are born, one of these is dominant, usually.
So in meiosis, the 23 chromosome one, that becomes 46 when that egg, if it's an egg, is fertilized and then goes.
Right, and that's what we're gonna, I think that's the next slide actually. Oh, well, we'll do that in a second. So basically this sperm or egg over here, it could have a brown eye allele. It might have a red hair allele and a short. The one here, maybe it's blue eyes, blonde hair, and tall. So basically any combination, this is just 3 traits. We've got, 26,000 genes. So you can think of all the combinations you can come up with. That's a good question. All right, so how is the end result of meiosis similar to the end result of mitosis then? If you're not sure, I would glance up at the board there where we drew it all out. I think there's 22, 23 people in here. All right, apparently I have some persuasive people in your group there. F is correct, none of the above. Which one were we deciding between? Did y'all put D? Oh yeah. So only in mitosis do you end up with 46 chromosomes. In meiosis, you end up with cells with 23. All right, so next thing you're going to do is using our fun little markers is you're going to make a concept map that shows the whole sexual life cycle. Look around. Let me know if you don't have any. Adriana, she stole them all. She has 6 of them. All right. So before you get started, let me tell you what how I view concept maps and how I would start this one. So basically, how I view them is I gave you a long list of words there on the assignment, and I would put each of those in a box. So I forgot what I put all on there. Is adult on there? All right, so adult, male. So it gets in a box. Any of those terms on there seem to match that one? Okay, so it would probably get connected to female at some point. Somebody else said sperm. And then is there any word on there that connects these? Eventually. What about how you make a sperm?
Oh, meiosis.
Okay, so maybe I put meiosis over here. I can never spell it. And you put an arrow, so it's like you're making a paragraph. So what word would go between adult male, blank, meiosis, blank sperm? Like a verb phrase. So it's a word that's not on there. Like just a verb phrase. You're making a sentence. Undergoes. That works. Is there an E on there? And then, so adult male undergoes meiosis, blank sperm. So how about to produce? So that's a good start. And I want you to put all those other terms in boxes and connect them all together on your table. So that way you can erase, start over, move things around. So we will be doing lots of these concept maps during the semester and the next semester. So they're really good for you to see how different words or processes are connected to each other. So a few things I saw from out there. First of all, a lot of these aren't like one straight pathway. There's stuff on the sides. There's arrows coming out, side, whatever. That's fine. And what's cool about these is it's however your brain thinks is what you're putting down there. So if you look around, everybody's is a little bit different, but they're all right. Some people went male and female down and then combined here. Some people started with the zygote and went out that way. But you basically all have the answer. So you're starting with adults. You're going through meiosis to make gametes, which are sperm and eggs. Sperm and egg meet through fertilization to make a zygote. Zygotes go through mitosis to make the adults. That's life cycles. All right. And the last thing, these concept maps do get easier to do as you do more of them. You can always take pictures of concept maps that you draw. You don't have to copy them down. You can always just take a picture of them. All right. So next up is the central dogma. The central dogma is talking about transcription and translation. And kind of the key thing about this is your DNA doesn't leave your nucleus. It stays there. It can never leave. But that's where all the information is to make all the proteins. So the proteins are what do the work of the cell, but they're made outside the nucleus. So you need some way to get the information that's in the nucleus outside the nucleus so you can use that information to make the proteins. That's what the messenger RNA is for.
So then, because of that, like with the plasmid membrane, that's how they get in and out? Or is that just like a permeable?
They go through the nuclear membrane.
Okay.
Yeah. I need a better eraser. All right, so let's say you're starting in your nucleus. You've got your DNA. So it's all your chromosomes in there. This is the nucleus. And then way outside, this is the cell membrane. What's this one? What membrane? This the nuclear membrane. What structure makes proteins? Ribosomes. Let's give her. And a lot of the ribosomes are at the rough ER. So we'll call this the rough ER. And maybe in green here. Just make a little circle. That's the ribosome. So you've got your ribosome out here. It makes the proteins. You've got your DNA in the nucleus. It's got all the information for how to make the proteins. So you need to connect them somehow. And the way they're connected is through mRNA. So in the nucleus, what's the first process that starts with the T again? So we go DNA to mRNA. And that was transcription. And then your mRNA leaves the nucleus and comes over here to the ribosome. And what happens to the mRNA? Gets turned into, so it's going to get turned into a protein at the ribosome. And what was that called? That's translation. So that's basically, that's the simple central dogma. That's what they call it. So DNA to mRNA in the nucleus, which is transcription. mRNA leaves, goes to the ribosome reads it, and creates a protein from those instructions.
How does the DNA get made into the mRNA?
Don't worry about it.
My brain does not work like that. That's what's so hard about this.
Yeah.
I'm like, oh, that's cool, that makes sense, but how did all that other stuff happen?
And that is what's hard about teaching the entire intro bio class in one chapter. Because if you were taking bio 111, that would be a whole chapter, how that happens. This would be a whole chapter over here. So if you want to watch, if you really have to know more, just type transcription animation into YouTube and watch a 10-minute movie on it. And it'll show you everything you want to know. Yeah, the book kind of goes a little crazy for some of these.
Went in so deep, and then I'm going, well, what's that? How'd that happen? Oh, well, what was that?
And the thing is, for this chapter, I was trying to pick out the things that I thought were most important for everything you're going to learn later. So like part 2 of the chapter, all that stuff about transport and osmosis, active transport pumps, that's stuff we keep using over and over again for A&P. This, we're probably not going to talk a whole lot about. But you should still know this is where your proteins come from that do everything. All right, and here's a little example movie too. If you're gonna watch that. How much time? Let's see. How much time do we have left? 25 minutes. Oh, but we still have the factory thing. 2. All right, so where is mRNA made? What do y'all think? Look at my chicken scratch up here. It's obvious.
Hey, at least it's in different colors.
I know. I try. All right, so 80% for D, which is correct. It is in the nucleus. All right, what about proteins? Where do you make proteins? All right, so before we get started on that, I realize there's something we could add to our diagram of our cell up here. So the ER sorts the proteins, decides where they need to go. Maybe they need to go to the mitochondria. Maybe that protein needs to go to the nucleus. So that's what's happening here.
Do you know what kind of proteins they are?
as to where they go?
Yeah, they go to where they're needed. So if they're a protein that makes ATP, they go to the mitochondria. If they're a protein that makes mRNA, they go to the nucleus.
So when they say synthesis, it just means it makes it.
Yeah, synthesis, a fancy word for makes.
Okay.
I know they love that word.
That clears up a whole lot of stuff.
Yeah,
I was right there.
that's a good.
I mean,
You know
I
what?
just looked it up, but
I'm gonna put that on the study guide for next semester. Synthesis. It's a good idea. Now, if a protein, if its job is like a hormone, it's supposed to leave. It goes from the ER to another little thing that's the Golgi. What color's our protein? I didn't make a protein. Yeah, let's make it a little triangle here. So there's our protein we made. It goes through the rough ER. It's not an A. It'll get to the Golgi, and the Golgi wraps it up nice like a little present in a vesicle, and then it goes to the cell membrane and gets released. So this is our protein that's going for export, or going to, like it's a hormone, or who knows what, it could be anything. So the rough ER sorts, the Golgi packages, and sends things out of the cell.
It's crazy how much it looks like so much, but it goes so fast. Like it happens how many times a day that happens, you know?
Oh yeah.
Like every minute, how much time?
So you've got, I don't know, probably 100,000 different proteins you can make in your cells. And different cells are making different ones, different times, different speeds.
and the lysosomes, like they use your hormones too, like the hormones that come in your cells, right?
The lysosome's more, it's the recycling
Or
center.
no, that's your Golgi does that too, though. Like they'll use them, right? And then
Yeah,
the
the
packages
Golgi,
go into the cell too, right?
right.
Okay.
But for now, just keep it a little simpler, but you are right. All right, so I'm going to practice this a little bit. And basically what you're practicing is you're going to do an analogy for your cell. So that's on the back side of this one. You're going to do this in pairs. And so your analogy is, because cells are like factories, so you're going to imagine that a cell is a factory. What would each of those things represent? All right, so thanks for playing my factory game. I promise it'll be helpful if you're stuck on the test or something, and it says Golgi apparatus. You're like, wait, what was the Golgi? What? You might think about your factory and be able to remember it. Might help. We'll finish this up next time. It's 2:20. So I'll see y'all on Monday for lab.